Influenza infection. It might start with a sneeze, but the underlying infection can cause far worse. Hundreds of thousands of people every year die from severe influenza virus infection. Everything begins when the virus enters our airways. Here, influenza viruses specifically attach to the surface of the epithelial cells. The viral membrane envelope contains the neuraminidase protein Na, important for the efficient release of newly produced viruses. The M2 ion channel promotes viral structural changes during cellular entry, and the influenza hemagglutinin protein HA, the key player for viral internalization, which facilitates viral binding to sialic acid decorated receptors and subsequently viral fusion. Proteolytic cleavage by host enzymes is critical for the activation of the HA trimers. Soluble or cell-bound host proteases cleave the precursor HA molecules into two parts, HA1 and HA2. Influenza virus particles are internalized into early endosomes by clathrin-mediated endocytosis. In late endosomes, the pH drops, triggering the conformational change of the cleaved HA molecules. HA1 opens up and allows HA2 to form a triple alpha helix bundle, which extends towards the endosomal membrane. Once the fusion peptides are anchored in the endosomal membrane, the whole molecule can fold back, allowing the fusion of the viral and endosomal membranes. After fusion, the viral genome can be released into the cytosol. The 8 viral RNA segments make their way into the cell nucleus, and the production of the new virus begins. Just hours after the initial infection, thousands of new viruses bud off the cell surface and infect neighboring cells. To stop the influenza infection, Crucell researchers have discovered fully human monoclonal antibodies capable of neutralizing the virus. These antibodies specifically bind to the HA and are internalized together with the virus. When the pH drops in late endosomes, the antibodies remain bound to a highly conserved epitope located in the stem of the HA. The antibodies now block the conformational change of HA, thereby preventing viral fusion and infection. The trapped virus degrades, leaving the cell unharmed. Some of Crucell's new antibodies can also prevent the initial cleavage of HA. They bind to a highly conserved epitope close to the HA cleavage site, thereby preventing host proteases from activating the virus. The uncleaved virus is not infectious, so the cell and patient are safe. Crucell's neutralizing antibodies prevent the spread of influenza infection and may save your life.
This animation will show how you get a cold. A cold is a viral illness which usually lasts for about 1 week. Click the navigation arrows below the animation screen to play, pause, rewind or fast forward the animation. Cold viruses, such as the rhinovirus and coronavirus, are taken into the body when you breathe them in from the air or when you touch surfaces which have the virus on them and then touch your mouth, nose or eyes. You can also get cold viruses from close or direct contact with someone with the virus, by kissing someone with the virus, for example. The eyes, nose, mouth and airways are all connected. Cold viruses can pass through these tubes and cavities to cause the symptoms of a cold. Cold viruses grow in soft, warm surfaces, such as your nose, throat, sinuses or airways. Infection with a cold virus causes the immune system to produce antibodies which attach to the virus and destroy it. Your body also makes mucus which traps the virus. You may swallow the viruses, so they're destroyed by the acid in your stomach, or they may be removed when you blow your nose. Excess mucus production causes the symptoms of a blocked nose, sneezing and coughing. You may also have a headache, which may be caused by the pressure in the sinuses blocked with mucus. Your body gradually fights the infection, so you feel better after about a week.
The staging of lung cancer refers to the extent of the disease. If you have the most common form of lung cancer, or non-small cell lung cancer, it will be staged according to the size of the tumor and whether it has spread to the lymph nodes or other organs of the body. In the occult, or hidden stage, cancer cells are found in coughed-up mucus or in other lung fluids, but no tumor can be seen in the lungs. Stage 0 is not considered invasive because the cancer cells are found in the innermost lining of the air passages but have not grown through it. Stage 1A is considered invasive cancer because the tumor has grown through the inner lining of the airways into the deep lung tissue. When measured, the tumor is no more than 3 centimeters in diameter and has not spread to the bronchi, lymph nodes, or distant sites. In stage 1B, the diameter of the tumor is larger than 3 centimeters, or has spread to a main bronchus, or has grown through the lung into the inner layer of the pleura. In stage 2A, the tumor is no more than 3 centimeters across, but it has spread to nearby lymph nodes on the same side of the chest as the tumor. There are 2 scenarios that can occur with stage 2B lung cancer. 1, where cancer cells have not spread to nearby lymph nodes, but have invaded the chest wall, diaphragm, pleura, the membranes surrounding the heart, or a main bronchus. In the second scenario for stage 2B, the cancer has spread to the lymph nodes on the same side of the chest as the tumor. Also, the tumor is larger than 3 centimeters across, or has grown into a main bronchus, or has grown through the lung into the pleura. In stage 3A, the tumor may be any size, and the cancer cells have spread to the lymph nodes on the same side as the lung tumor. Also, the cancer may have spread to the chest wall, diaphragm, pleura, the membranes around the heart, or a main bronchus, but not the trachea. In stage 3B, the tumor may be any size and has spread to lymph nodes above the collarbone or on the other side of the chest. The cancer may have spread to nearby structures, such as the heart, major heart vessels, diaphragm, chest wall, trachea, esophagus, breastbone, or to more than one place in the same lung. In stage 4, cancer has spread to other lobes of the same lung or into the opposite lung and has spread to other parts of the body, such as the brain, liver, kidneys, adrenal glands, or bone. Treatment options for cancer include local therapies, such as surgery and radiation therapy, and systemic therapies, such as chemotherapy and targeted therapy. You may receive one or a combination of these treatments. There are several surgical options, depending on the type and stage of the cancer. During a wedge resection, or segmenectomy, the tumor and a small margin of healthy tissue are removed. In a lobectomy, or sleeve lobectomy, one lobe of a lung is removed. During a pneumonectomy, the surgeon removes the entire lung. In a sleeve resection, a diseased section of a large airway is removed, and the healthy ends are reattached. During any of these procedures, your surgeon may remove lymph to check them for signs of cancer. Radiation therapy, or radiotherapy, is a local treatment that uses high-energy rays to kill or shrink cancer cells to relieve symptoms. External beam radiation therapy is directed at the cancer's location from a machine outside your body. Brachytherapy, or internal radiation therapy, often in the form of pellets, is most commonly used to help relieve blockage of the large airways by cancer. Chemotherapy is a systemic treatment that uses drugs to stop the growth of the cancer cells by either killing them or inhibiting their cell division. Once the drugs enter the bloodstream, they can travel and reach cancer cells throughout the body. Targeted therapies are newer cancer treatments that work by focusing on specific genetic abnormalities of cancer cells. One targeted therapy, bevacizumab, starves the tumor by stopping it from creating a new blood supply. Another targeted therapy, erlotinib, stops tumor growth by blocking the chemicals that signal the cancer cells to grow and divide.